Hi, this is Tiffany Tula, week 6, Bio 225. This is our week 6 lab of environmental influences on microbial growth, salt tolerance testing. I'm going to be performing the first parts of the test before inoculation, or the inoculation parts actually, and then I'll incubate for about 48 hours and then take a look at them. I've cleaned my work area, washed my hands, put my gloves on. I have everything sitting in alcohol to sterilize, and I've labeled all my tubes accordingly. And then here's the other ones for the second portion of the test. I have my hydrochloric acid, and I'll be doing that portion as well. Let's get to the inoculation. I'm going to start with the Estervacier broth. I've already sterilized my pipette, shaking it dry. I'm going to flame the lid as always. This is the 1% NaCl broth. Going to inoculate first. I'm gonna be adding 2 drops of the active Estervacier culture broth into each tube. So flame the lid before I add the drops. I'm going to add 2 drops. 1, 2. Flame the lid. Add it back on, cap. Now I'm going to do the same with the 7% broth, as you can see. Flaming the lid. Adding 2 drops. Let's get some more in here. 1, 2. And the lid. And then lastly, with the 15% broth. No, 1, 2, and lid. Place the cap, and I'm going to continue doing that same procedure with the S. epidermis broths. Let's flame this lid before I replace the cap. And I'll continue on to the next microorganism. So right now I'm going to do the second portion with the HCl. I have my pH of 5 tubes. Going to flame the top and drop exactly 1 drop into the pH 5. I'm going to flame it and replace it. I'm going to do the same. That was for my S-Epi. Now I'm going to do the same for my Estervacier tube. Let's see. Exactly 1 drop. Flame again. Place the cap, and since the pH is already 7 for the broths, I don't have to do anything to those, and I'll move on to the pH of 9. Now doing the same with the pH of 9 tubes, I have my sodium hydroxide that I will be using. Flame this tube. I'll be adding 1 drop into tube, flaming it again, replacing the lid, and I'll do that the same for the S-Epi tube. So now that my tubes are prepared with the change in pH, I'm going to inoculate them with the active cultures. So let's go through this. It'll be S. epidermis first. Let's see here. So I always sterilize the top. This is my pH of 5 tube. I'll take 2 drops and place it inside. Flame, place the cap back on, and I'll continue doing that for the rest of the tube. Next, moving on to my pH of 7. I'll do the same for the S. cerevisiae tubes, and then I'll place them in my incubation area for 48 hours. So I've let my microbes incubate for 24 hours, and now we're going to take a look at them in the different levels of pH to see the change in possibly turbidity. Here you can see my S-Epi tubes. There is turbidity present. And if we move on to the S. cerevisiae tubes, there is turbidity as well. The tops of these have a little bit of flaky growth, while these do not. I'm gonna take a picture, record it, and put it in my lab report. It's been 48 hours, so now I'm gonna take a look at my tubes that are being tested for the salt content and take a look at the growth that happened in them. As you can see, this is the S. cerevisiae tubes. There is a significant difference in turbidity between the first tube with only 1% and the last tube with 15%. And moving on to the S-Epi, there is also a significant difference in turbidity. That's with the 1%. And this is, you can see the flux in there. It's with the 7% and practically clear with the 15%. So I'm gonna take pictures and log this into my lab report and then turn it in.